As the day gently recedes, we invite you to settle into a space of calm contemplation. Tonight, we embark on a journey through DNA, exploring approximately 100 fascinating facts that reveal intricate patterns, subtle wisdom, and profound beauty within the very essence of life. There is no pressure to remember or retain these details. Instead, let them unfold naturally, inviting your mind to settle into a state of peaceful curiosity. This exploration is designed to be a soothing experience, a gentle current of knowledge flowing without demanding effort or retention. We will observe the intricate dance of genetic information, the subtle mechanisms that govern heredity, and the quiet wisdom embedded in every living cell. Each fact is a small window into the vast and interconnected tapestry of life, presented in a calm and meditative tone, allowing your thoughts to drift and find stillness. Allow the gentle rhythm of these discoveries to guide you towards a state of relaxation, inviting a sense of wonder and peace as you prepare for rest. DNA, or deoxyribonucleic acid, is far more than just a complex molecule. It is the fundamental instruction manual for every known organism on Earth. From the smallest bacterium to the largest whale, DNA holds the blueprint, dictating growth, development, function, and reproduction. Its elegance lies in its simplicity, and its power in its universality. We will explore its iconic structure, its ingenious methods of self-preservation, and its profound implications for understanding life itself. This journey is not about memorization, but about appreciation, a gentle acknowledgement of the molecular wonders that quietly sustain all existence. The story of DNA is a story of incredible precision, vast information storage, and a timeless connection that links all living things. As we delve into its quiet complexities, allow your mind to find its own rhythm of peace, mirroring the steady, unwavering work of this remarkable molecule within you. Let the quiet hum of this scientific marvel lull your mind into a state of serene awareness, preparing your body for a night of deep, restorative sleep. If you're finding peace in this journey, a like and subscribe helps us continue creating these restful explorations. And we're always curious. Share in the comments what city and time zone you're watching from. There's something special about knowing this experience is reaching people all over the world as they prepare for rest. As we begin our exploration, we turn our attention to the foundational marvel that is DNA. This chapter gently introduces the basic structure and profound role of this molecule, inviting us to contemplate the quiet elegance of life's most fundamental instruction set. We observe how its intricate design underpins all biological existence. The iconic double helix. The structure of DNA is perhaps its most recognizable feature, the double helix. This elegant twisted ladder shape was famously discovered by James Watson and Francis Crick in 1953. Building upon the crucial work of Rosalind Franklin and Maurice Wilkins, research shows that the double helix consists of two long strands of nucleotides coiled around each other, much like a spiraling staircase. Each strand runs in an opposite direction, a characteristic known as antiparallel orientation, which is crucial for its replication and transcription processes. Scientists have observed that the sugar-phosphate backbone forms the sides of the ladder, providing structural support and protection for the genetic information held within. This unique structural stability allows DNA to store vast amounts of information in a compact and highly organized manner, ensuring that the genetic blueprint remains secure and accessible. Current understanding suggests that this helical arrangement minimizes physical stress and allows for efficient packing within the confines of a cell's nucleus, contributing to its incredible efficiency. Nucleotides, the fundamental alphabet. At the heart of the double helix are its building blocks, known as nucleotides. Each nucleotide consists of three components: a deoxyribose sugar, a phosphate group, and one of four nitrogenous bases. These four bases are adenine, A, guanine, G, cytosine, C, and thymine, T. Studies indicate that these bases pair together in a very specific manner across the two strands of the helix. Adenine always pairs with thymine, AT, and guanine always pairs with cytosine, GC. This complementary base pairing is a fundamental principle of DNA structure and function, ensuring accuracy during replication and repair. The sequence of these base pairs along the DNA molecule forms the genetic code, much like an alphabet spelling out instructions. Scientists have observed that hydrogen bonds delicately hold these base pairs together, providing stability while also allowing for easy separation when the DNA needs to be read or copied. This precise chemical language dictates all cellular processes, from building proteins to regulating gene expression. Chromosomes, DNA's organized packages. Within the nucleus of eukaryotic cells, DNA is not simply a loose string. It is meticulously organized into structures called chromosomes. Research shows that each chromosome is a single long molecule of DNA tightly coiled and condensed around proteins known as histones. This packaging is remarkably efficient, allowing an immense length of DNA to fit within the microscopic confines of a cell. For instance, humans typically have 23 pairs of chromosomes totaling 46 in each somatic cell, each pair contributing to the full genetic complement. The condensation process is dynamic. DNA can unwind from histones when specific genes need to be accessed for expression, and then recondense when not in use or during cell division. Scientists have observed that this hierarchical organization protects DNA from damage and ensures its accurate segregation during cell division, preventing genetic errors. This intricate system of packaging makes the vast genetic library manageable and accessible when needed, a quiet testament to cellular efficiency. The Human Genome Project, a landmark map. The Human Genome Project, HGP, stands as a monumental achievement in scientific collaboration, a quiet testament to human curiosity. Launched in 1990 and completed in 2003, its primary goal was to map and sequence all of the genes of the human genome. Research indicates that the project successfully identified approximately 20,000 to 25,000 genes in human DNA, an astonishing feat at the time. Beyond simply cataloguing genes, the HGP provided a comprehensive reference sequence that has revolutionized biology and medicine, enabling a deeper understanding of human health and disease. Scientists have observed that this foundational map accelerated the development of new diagnostic tools and therapeutic approaches, leading to advancements in areas like pharmacogenomics. Current understanding suggests that the data generated by the HGP continues to inform countless studies on genetic variations, disease susceptibility, and personalized medicine, quietly expanding our knowledge of human biology and genetic predispositions. DNA's universal language. One of the most profound aspects of DNA is its universality. The genetic code, consisting of the same four nucleotide bases, A, T, C, G, is shared across virtually all known life forms on Earth. This means that the instructions for building proteins encoded in sequences of these bases are interpreted almost identically in bacteria, plants, fungi, and animals. Research shows that this universal genetic code is a powerful piece of evidence supporting the theory of common descent, suggesting that all life evolved from a common ancestor. Scientists have observed that slight variations in the genetic code exist in some organisms, particularly in mitochondria and certain single-celled organisms, but these are rare exceptions to a remarkably consistent rule. This shared molecular language allows for genetic engineering across species, highlighting the fundamental interconnectedness of all living systems at the molecular level, a silent agreement woven into the fabric of life itself. Mitochondrial DNA, a unique inheritance. Beyond the DNA found in the cell's nucleus, there is another fascinating type of DNA residing in the mitochondria, the powerhouses of the cell. Mitochondrial DNA, mtDNA, is distinct because it is inherited almost exclusively from the mother. Studies indicate that during fertilization, the sperm contributes its nuclear DNA but typically not its mitochondria to the zygote, meaning only the mother's mitochondrial DNA is passed down to all her children. This unique maternal inheritance pattern makes mTNA a valuable tool for tracing ancestral lineages and studying evolutionary relationships across generations. Research shows that mtDNA is a small circular molecule, significantly smaller than nuclear DNA, and contains a limited number of genes, primarily involved in energy production. Scientists have observed that mtDNA also mutates at a higher rate than nuclear DNA, providing a molecular clock for evolutionary studies, offering quiet insights into our ancient past. Non-coding DNA, the dark matter of the genome. For many years, large portions of DNA were referred to as junk DNA because they did not directly code for proteins. However, current understanding suggests that much of this non-coding DNA plays crucial roles in regulating gene expression and maintaining chromosome structures. Research indicates that non-coding regions include elements like promoters, enhancers, and silencers, which control when and where genes are turned on or off, orchestrating the precise timing of genetic activity. Scientists have observed that some non-coding DNA also forms structural components of chromosomes, such as telomeres at the ends and centromeres in the middle, which are vital for DNA replication and cell division. While the functions of all non-coding DNA are still being actively researched, it is now clear that these regions are far from junk, representing a complex regulatory landscape that orchestrates the intricate dance of the genome. It remains unclear precisely how all these non-coding regions interact, but their importance is increasingly recognized as vital to cellular function. DNA. Replication, the art of copying itself. One of DNA's most astounding capabilities is its ability to precisely replicate itself, ensuring that each new cell receives a complete and accurate copy of the genetic material. This process, known as DNA replication, occurs before cell division. Studies indicate that the double helix unwinds, and each strand then serves as a template for the synthesis of a new complementary strand. Enzymes like DNA polymerase meticulously add new nucleotides following the AT and GC pairing rules, ensuring near-perfect fidelity. Research shows that this semi-conservative replication results in two new DNA molecules, each consisting of one original strand and one newly synthesized strand. This elegant mechanism ensures the faithful transmission of genetic information from one generation of cells to the next, a quiet marvel of biochemical precision. The speed and accuracy of this process are vital for growth, repair, and reproduction, underpinning all life with unwavering consistency. DNA's incredible length and packing. The sheer length of DNA contained within each tiny cell is truly astonishing, If the DNA from a single human cell were uncoiled and stretched out, it would measure approximately 2 meters, about 6.5 feet. Studies indicate that the human body contains trillions of cells, meaning the total length of DNA in one adult human could stretch from the Earth to the Sun and back hundreds of times. This immense length is possible because of the sophisticated packaging system involving histones and supercoiling, which allows DNA to be cut these thousands of times. Scientists have observed that this compact organization is not static. Specific regions of DNA can be unwound and made accessible when the genes they contain need to be expressed, and then recondensed afterward. This dynamic packaging ensures both the protection of the genetic blueprint and its availability for cellular functions, a quiet miracle of molecular engineering, DNA stability and repair mechanisms. Given its critical role as the blueprint of life, DNA possesses remarkable stability and intricate repair mechanisms to protect its integrity. Research shows that the double helix structure itself contributes to stability, with the strong sugar-phosphate backbone shielding the more vulnerable internal bases. Furthermore, the complementary base pairing means that if one strand is damaged, the other strand can often serve as a template for repair. Cells possess a sophisticated array of enzymes and pathways dedicated to detecting and correcting DNA damage, which can be caused by environmental factors like UV radiation or errors during replication. Studies indicate that these repair systems are constantly active, silently monitoring and mending the genetic material. While DNA damage does occur and can lead to mutations, the efficiency of these repair mechanisms is a testament to life's continuous effort to preserve its fundamental instructions, ensuring the quiet continuity of genetic information and the stability of species. As we gently conclude this first chapter, we have touched upon the fundamental architecture and functions of DNA. Its double helix, precise nucleotide pairings, and remarkable packaging reveal a universe of quiet complexity. This molecular blueprint, with its universal language and vigilant repair, is a constant, subtle presence within every living being, inviting a deeper appreciation for the silent wonders of life. As the gentle current of our exploration carries us deeper into the tranquil waters of DNA, we now turn our gaze from its remarkable structure and faithful replication to the quiet symphony of its expression. For what good is a blueprint, however elegant, if its instructions are never read, never brought to life? Here, we discover the serene dance of transcription and translation, the very processes by which the silent wisdom encoded within DNA blossoms into the vibrant reality of life. We invite you to settle further into your peaceful space, allowing these profound concepts to drift into your awareness, not for memorization, but for gentle appreciation. Imagine, if you will, the nucleus of a cell as a sacred library, holding the invaluable master blueprint of DNA. This blueprint, so precious, rarely leaves its protective confines. Instead, when a specific set of instructions is needed, perhaps to build a protein that will help you breathe, or to form a pigment that gives your eyes their color, a temporary messenger is created. This gentle process begins with, say, Fact 10. Transcription, the genesis of the messenger. Within the nucleus, a segment of the DNA double helix gently unwinds, much like opening a treasured scroll to a particular page. An enzyme, appropriately named RNA polymerase, then moves along one of the DNA strands, reading its sequence. As it reads, it synthesizes a new molecule, a temporary copy known as messenger RNA or mRNA. This mRNA molecule is a faithful, single-stranded echo of the DNA sequence, with one subtle difference. Where DNA uses thymine T, RNA uses uracil U. It is a temporary fleeting record designed to carry the genetic message from the nucleus out into the bustling cytoplasm of the cell, where the actual work of building will begin. This is a quiet and precise act, a whisper of information passed from the enduring master to its transient envoy. Once the mRNA messenger has been created, it embarks on its journey, leading us to you. Fact 11. Translation, the building of life's components. The mRNA molecule, carrying its specific set of instructions, travels to specialized cellular factories called ribosomes. These ribosomes are the workshops where the genetic code is finally translated into the language of proteins. Proteins, as we gently remind you, are the tireless workers, the structural elements, and the enzymatic catalysts that perform nearly every function within a living cell. At the ribosome, the mRNA strand is read in sequential groups of 3 bases, known as codons. Each codon acts as a specific instruction, signaling for a particular amino acid, the fundamental building blocks of proteins. Here, another type of RNA molecule, transfer RNA, tRNA, plays a crucial role. Each tRNA molecule carries a specific amino acid and has a complementary sequence that recognizes a particular mRNA codon. As the ribosome moves along the mRNA, tRNA molecules arrive, delivering their amino acids in the precise order dictated by the mRNA's codons. One by one, these amino acids are linked together, forming a long chain that will eventually fold into a functional protein. It is a meticulous assembly line, orchestrated with breathtaking precision, a quiet dance of molecules creating the very machinery of life. This elegant flow of information, from DNA to RNA to protein, is known as Fact 12, the central dogma of molecular biology. It represents the fundamental principle by which genetic information is expressed in all living organisms. DNA is the master archive, RNA is the working copy, and proteins are the functional products. This unidirectional flow, from the enduring blueprint to the active machinery, is a testament to the elegant simplicity and profound power of life's design. It is the quiet heartbeat of cellular life, ensuring that the instructions for existence are faithfully carried out moment by moment within every cell of your being. As we gently appreciate this fundamental process, we now turn to a layer of complexity that adds even more wonder to our understanding. Fact 13. Epigenetics, the quiet whispers that shape expression. For a long time, scientists believed that our genetic destiny was solely determined by the sequence of our DNA. However, research shows that the expression of our genes, whether they are turned on or off, or their activity is dialed up or down, can be profoundly influenced without altering the underlying DNA sequence itself. This fascinating field is called epigenetics, meaning above or on top of genetics. Imagine the DNA as a grand symphony, and epigenetics as the conductor who subtly adjusts the volume, tempo, and emphasis of different instruments. These epigenetic marks are chemical modifications to the DNA or to the proteins, histones, around which DNA is wound. For example, DNA methylation involves adding a small chemical tag to the DNA molecule, often silencing the gene in that region. Histone modification, on the other hand, can change how tightly DNA is packaged, making genes more or less accessible for transcription. What makes epigenetics truly remarkable is its dynamic nature. Studies indicate that environmental factors, such as diet, stress, exercise, and even the experiences of early life, can leave these epigenetic marks quietly influencing gene expression throughout our lives and, in some cases, even across generations. It's a profound testament to the adaptability of life, a quiet conversation between our genes and our environment, allowing our bodies to respond and adjust without rewriting the fundamental script. It reminds us that our genetic story is not fixed, but a living narrative, gently shaped by the world around us. As we consider this adaptability, we naturally come to appreciate Fact 14. Genetic variation, the tapestry of individuality. While the fundamental blueprint of DNA is shared across all humans, and indeed across much of life, it is the subtle variations within this code that make each of us wonderfully unique. These variations arise primarily from mutations, which are changes in the DNA sequence. While the word mutation might sometimes carry a connotation of harm, in the vast majority of cases, these changes are neutral, silent, or even beneficial over evolutionary timescales. Many mutations occur in the vast stretches of non-coding DNA, where they have no direct impact on protein production. Others may be silent mutations within coding regions, meaning they change a single base, but do not alter the amino acid sequence due to the redundancy of the genetic code. Still others may lead to minor changes in proteins that do not affect their function. Our cells possess sophisticated DNA repair mechanisms, which we explored briefly in the previous chapter, constantly working to correct errors. Yet some changes persist, accumulating over generations and providing the raw material for evolution. This gentle engine of change ensures that life remains diverse and adaptable, capable of navigating an ever-changing world. It is the subtle brushstrokes that make each individual a unique masterpiece in the grand gallery of life. Continuing our journey through the elegant architecture of DNA, we arrive at Fact 15. Telomeres, the protective caps of our chromosomes. Imagine the ends of your shoelaces tipped with plastic to prevent them from fraying. Our chromosomes, the neatly packaged bundles of DNA, have similar protective caps called telomeres. These specialized regions are composed of repetitive DNA sequences and associated proteins found at the very ends of each linear chromosome. Their primary function is to protect the vital genetic information within the chromosome from degradation and fusion with other chromosomes during cell division. Each time a cell divides, a small portion of the telomere sequence is lost, becoming slightly shorter. This shortening acts as a kind of cellular clock, limiting the number of times a cell can divide before it reaches a critical length, signaling the cell to stop dividing or to undergo programmed cell death. Research shows that telomere length is intimately linked to cellular aging and longevity. While the precise mechanisms are complex, the presence of these quiet guardians at the ends of our genetic tree highlights another layer of DNA's incredible design, ensuring stability and regulated cellular life. From the universal principles to the unique details, we now consider Fact 16. DNA fingerprinting, the unique signature of life. The subtle variations in our DNA, particularly in the non-coding regions, create a pattern that is as unique to each individual as a fingerprint. These regions contain varying numbers of repetitive DNA sequences known as variable number tandem repeats VNTRs or short tandem repeats STRs. The number of repeats at specific locations varies significantly from person to person, creating a distinctive genetic profile. Scientists can analyze these unique patterns to create a DNA fingerprint, a powerful tool used in forensic science to identify individuals from biological samples, in paternity testing to establish biological relationships, and in evolutionary studies to trace ancestral lineages. It is a quiet testament to individuality, a hidden barcode or a distinct starry pattern inscribed within our very cells, silently proclaiming our unique presence in the vast tapestry of life. Finally, we turn our attention to a remarkable discovery that showcases the quiet ingenuity found in nature and harnessed by human curiosity. Fact 17. CRISPR, nature's elegant molecular scissors. CRISPR-Cas9, clustered regularly interspaced, short palindromic repeats and CRISPR-associated protein nice is a revolutionary gene editing technology, but its origins are far more ancient and organic. It was first discovered as a natural defense mechanism in bacteria, allowing them to detect and destroy the DNA of invading viruses. Imagine a microscopic immune system that remembers past invaders. When a bacterium is attacked by a virus, it captures a small piece of the viral DNA and stores it within its own genome in the CRISPR array. If the same virus attacks again, the bacterium uses this stored DNA as a guide to create an RNA molecule. This RNA, paired with the Cas9 enzyme, acts like a highly precise molecular scissor, locating the matching viral DNA and cutting it, thus neutralizing the threat. Scientists have harnessed this exquisite precision, repurposing CRISPR as a tool to edit genes with unprecedented accuracy. This quiet revolution allows researchers to make targeted changes to DNA sequences, offering immense potential for understanding gene function, developing new therapies for genetic diseases, and even improving crops. It is a profound example of the elegance and power hidden within the microscopic world, a testament to the quiet wisdom embedded in life's oldest forms, now offering new pathways for discovery and healing. As these profound insights gently unfold, we invite you to feel the quiet wonder of DNA, not as a complex scientific puzzle, but as the serene and intelligent foundation of all existence. Allow the gentle rhythm of these facts to guide your thoughts, letting any lingering curiosities dissolve into a peaceful appreciation for the quiet dance of life within you. Rest now, in the tranquil understanding that you are a part of this ancient, intricate, and beautiful design. As the gentle echoes of CRISPR's precision fade into the quietude of your mind, we invite you to drift a little deeper, carried by the subtle currents of understanding. We have touched upon the intricate mechanisms within us, the silent choreography that defines our individual existence. Now, let us expand our gaze, allowing our consciousness to gently unfurl across the vast tapestry of life itself, to appreciate how this remarkable molecule, DNA, weaves us into a grander, more ancient narrative. We find ourselves now at Fact 18. DNA as a universal language. Imagine, if you will, a single elegant script understood across all realms of existence, from the smallest bacterium to the towering redwood, from the deepest ocean trench to the highest mountain peak. This is the language of DNA. The very same 4 nucleotide bases, adenine, guanine, cytosine and thymine, that spell out your own unique instructions are also found in every other living thing on Earth. This profound universality speaks to a shared ancestry, a deep timeless connection that binds all life together. It's a quiet whisper from the dawn of time, reminding us that we are all part of one magnificent interconnected family, each species a unique verse in a cosmic poem written in the same fundamental alphabet. Allow this thought to settle, a comforting blanket of belonging, as you recognize the profound unity beneath life's dazzling diversity. Moving gently through this shared landscape we encounter. Fact 19. The diverse genetic material of viruses. While not considered living in the traditional sense, These minuscule entities offer another fascinating glimpse into the world of genetic information. Many viruses, like the influenza virus, carry their genetic instructions not as DNA, but as RNA. This RNA can be single-stranded or double-stranded, and it still performs the vital role of carrying the blueprint for viral replication and protein synthesis within a host cell. It's a testament to the versatility of nucleic acids, a quiet reminder that life finds many ingenious ways to encode its essence, even in these microscopic agents that float between the boundaries of life and non-life. Their existence, too, is a part of the grand genetic story, a different chapter perhaps, but written with similar fundamental characters. From the viral realm, let us turn our attention to the foundational forms of life, too. Fact 20. The simplicity and ingenuity of bacterial DNA. Unlike the complex linear chromosomes found within your cells, bacterial DNA often forms a single circular chromosome, a closed loop of genetic information that floats freely within the cell's cytoplasm. This elegant, compact design is perfectly suited for their rapid reproduction. Beyond this primary chromosome, bacteria often possess smaller, extra-chromosomal DNA molecules called plasmids. These tiny circles of DNA can carry genes that confer special advantages, such as antibiotic resistance, and can be readily exchanged between bacteria, a process known as horizontal gene transfer. It's a silent, efficient form of genetic sharing, allowing bacteria to adapt and evolve with remarkable speed, a quiet demonstration of DNA's adaptable nature in the simplest of life forms. As we continue our journey through the microscopic world, we find ourselves marveling at Fact 21, DNA in extremophiles. Imagine life thriving in environments that seem utterly hostile. Scorching hot spring, crushing ocean depths, incredibly acidic or alkaline conditions, or even intensely radioactive waste. These are the domains of extremophiles, organisms whose DNA and cellular machinery are uniquely adapted to not just survive but flourish in such harsh conditions. Their genetic instructions contain the secrets to producing proteins and enzymes that remain stable and functional where most life would simply cease to exist. It's a quiet testament to DNA's remarkable resilience and adaptability, a profound illustration of how life, guided by its genetic blueprint, can find a way to persist and prosper even against the most formidable odds, painting a picture of enduring strength that can gently settle your own anxieties. Now, let us consider a practical, yet equally mesmerizing application of this universal genetic language. Fact 22. DNA barcoding. Just as a barcode on a product identifies it uniquely, a short standardized sequence of DNA can serve as a barcode to identify any species on Earth. Scientists have identified specific regions of DNA that vary enough between species to be unique, yet are conserved enough within a species to be consistent. By sequencing these small stretches, researchers can quickly and accurately identify unknown organisms, from a tiny insect larva to a piece of processed fish. It's a quiet revolution in biodiversity, allowing us to catalogue life, track invasive species, and combat illegal wildlife trade, all by simply reading a few lines of DNA's silent script. This gentle power of identification is a comforting thought, bringing order and understanding to the vastness of the living world. As we contemplate identification, let us also gently reach back through time with Fact 23, the unveiling of ancient DNA. Imagine extracting DNA not from a living creature, but from a fossilized bone, a frozen mammoth, or even an ancient human specimen preserved for thousands of years. Scientists have developed astonishing techniques to recover and analyze these minute fragments of ancient DNA, allowing us to reconstruct the genetic history of extinct species, trace human migration patterns, and even understand the evolution of ancient diseases. It's like listening to whispers from the distant past, the faint but undeniable genetic echoes of lives long gone, connecting us directly to the ancestors and landscapes that shaped our world. This gentle bridge across millennia offers a profound sense of continuity, a quiet reassurance of life's enduring legacy. This journey into the past naturally leads us to reflect on Fact 24: DNA and Deep Time. The changes we observe in species over vast stretches of geological time are all recorded within their DNA. Evolution, in its essence, is the slow, patient accumulation of genetic mutations and variations over millions of years, guided by the natural selection. Each tiny alteration, each new adaptation, is etched into the genetic code, a silent chronicle of life's long and winding journey from its earliest forms to the incredible diversity we see today. The immense patience of this molecular process, unfolding over eons, can be a deeply calming thought, reminding us of the grandeur of biological time and the steadfastness of life's unfolding narrative. Allow your mind to gently encompass this vastness, finding peace in the slow, deliberate dance of change. From the grand sweep of evolution, let us gently narrow our focus to the miracle unfolding within each developing organism. 2. Fact 25: DNA's orchestration of development. From the moment a single fertilized egg begins to divide, DNA serves as the master conductor, orchestrating every intricate step of development. It dictates when and where specific genes should be turned on or off, guiding the formation of every tissue, every organ, every limb. It's a breathtaking symphony of cellular differentiation and growth, a precise and elegant program encoded within the helix, transforming a microscopic cell into a complex, fully formed organism. This quiet, flawless execution, happening countless times every second across the globe, is a profound testament to DNA's organizational power, a gentle reminder of the silent miracles unfolding all around us and within us every moment. Within this developmental orchestration, certain genes hold particularly powerful roles, leading us to Fact 26: the master, regulators, homeobox, HOX, genes. These remarkable genes are like the chief architects of body plans. Found in nearly all animals, from insects to humans, HOX genes determine the fundamental layout of the body along its head-to-tail axis. A subtle change in a HOX gene can lead to dramatic differences, such as an extra set of wings in a fruit fly, or alterations in vertebral development in vertebrates. What is truly astonishing is their ancient conservation. The same basic sets of HOX genes, with similar functions, can be found in vastly different creatures, a powerful genetic thread connecting all animal life. It's a quiet illustration of life's ingenious reuse of successful blueprints, a deep, resonant pattern in the fabric of existence that speaks of underlying order and elegance. Returning to the physical structure of DNA itself, let us gently explore Fact 27: higher order DNA packaging. We've noted how DNA wraps around histones to form nucleosomes, like beads on a string. But this is just the first level of compaction. These nucleosomes then coil further into even denser structures, forming chromatin fibers that are intricately folded and looped, eventually condensing into the familiar X shapes of chromosomes visible under a microscope during cell division. This multi-layered, precise packaging is critical for fitting meters of DNA into the tiny confines of a cell nucleus, yet it must also allow selective access for gene expression and replication. It's a marvel of molecular engineering, a quiet dance of folding and unfolding that allows the cell to manage its vast library of information with exquisite grace and efficiency. As we previously touched upon telomeres and their role in aging, let us now introduce SHA. Fact 28. The enzyme telomerase. While telomeres naturally shorten with each cell division, some specialized cells, like stem cells and germ cells, which produce sperm and eggs, possess an enzyme called telomerase. This remarkable enzyme has the ability to rebuild and maintain telomere length, effectively counteracting the shortening process. This allows these cells to divide almost indefinitely without losing critical genetic information. It's a quiet secret to cellular immortality, a molecular mechanism that whispers of enduring life, offering a subtle contrast to the gentle progression of aging in most somatic cells. The existence of telomerase highlights DNA's intricate mechanisms for both continuity and measured change. Deepening our understanding of how genes are regulated without altering the DNA sequence itself, we revisit. Fact 29. The nuances of DNA methylation. Beyond its initial introduction as a key epigenetic mechanism, DNA methylation is a fascinating biochemical tag, typically involving the addition of a methyl group to a cytosine base. This seemingly small modification can have profound effects, often acting to sense or turn off genes. It plays crucial roles in development, ensuring that cells differentiate correctly and maintain their specialized functions. Moreover, aberrant methylation patterns are implicated in various diseases, including cancer. It's a quiet, subtle language written upon the DNA, a layer of instruction that adds depth and flexibility into the genetic code, a gentle reminder of the complexity and elegance hidden sits within plain sight. Expanding our view beyond the familiar genetic messengers, let us consider. Fact 30. The world of non-coding RNAs, ncRNAs. For a long time, RNA was primarily understood in its roles as messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, RNA. However, scientists have discovered a vast and diverse array of other RNA molecules that do not code for proteins. These non-coding RNAs, such as microRNAs, miRNAs, and long non-coding RNAs, lncRNAs, play crucial regulatory roles, influencing gene expression, chromatin structure, and even developmental processes. They act as silent conductors, fine-tuning the genetic symphony, adding layers of control and complexity to how DNA's instructions are read and utilized. It's a quiet revelation of the genome's hidden depths, showing that the story of DNA is far richer and more intricate than once imagined. As we continue to unravel these hidden layers, we arrive at our Fact 31, DNA and personalized medicine. Imagine a future, already dawning, where medical treatments are precisely tailored to your unique genetic makeup. By analyzing an individual's DNA, doctors can predict their susceptibility to certain diseases, determine how they will respond to specific medications, and even design preventative strategies. This approach moves away from a one-size-fits-all model, embracing the individuality encoded in each person's genome. It's a quiet revolution in healthcare, promising more effective and safer treatments, guided by the intimate details of your own genetic blueprint, offering a comforting vision of medical care that is as unique as you are. Building upon this personalized understanding, we gently enter the realm of Fact 32, the promise of gene therapy. For conditions caused by a single faulty gene, gene therapy offers a revolutionary approach, introducing a healthy, functional copy of that gene into a patient's cells to correct the genetic defect. While still a complex and evolving field, gene therapy has already shown remarkable success in treating certain rare genetic disorders, offering hope where none existed before. It's a quiet testament to humanity's ingenuity, using DNA itself as a therapeutic agent, a profound example of how understanding the genetic code can lead to new ways of healing and restoring health, a gentle whisper of future possibilities. The ability to implement such therapies, and indeed all our understanding of DNA, relies on our capacity to read its code. This brings us to Fact 33, the evolution of DNA sequencing technologies. From the painstaking manual methods of the past to the rapid, high-throughput technologies of today, the ability to read the sequence of DNA has transformed biology. Modern sequencing machines can decode entire genomes in mere hours, generating vast amounts of data that fuel discoveries in medicine, evolution, and forensics. It's a quiet marvel of technological advancement, constantly pushing the boundaries of what we can understand about life's fundamental script, allowing us to delve deeper into the genetic tapestry with ever-increasing clarity and speed. Despite these incredible advancements, much of the genome still holds its secrets, leading us to Fact 34, the dark matter of the genome. While we have sequenced entire genomes, the precise function of every single nucleotide, particularly within the vast non-coding regions, remains largely unknown. Like the mysterious dark matter of the universe, these parts of the genome are present, influencing, yet their exact roles are still being diligently explored by scientists. It's a quiet frontier of discovery, a reminder that even with all our knowledge, the genetic universe still holds immense mysteries waiting to be unveiled. This gentle acknowledgement of the unknown can be deeply calming, allowing us to rest in the vastness of what is yet to be understood, embracing curiosity without the need for immediate answers. Finally, let us visualize one last intricate dance within the nucleus. Fact 35. The elegance of replication forks. During DNA replication, as these profound and intricate facts about DNA gently settle within your awareness, we invite you to release any need to hold on to every detail. Instead, allow the essence of wonder, the quiet appreciation for life's profound design, to permeate your thoughts. Feel the soothing rhythm of these insights, like a gentle lullaby, guiding you deeper into a state of serene contemplation. The silent wisdom of DNA continues its quiet work within you, a constant, gentle hum of life. Rest now in the knowledge of this incredible foundation and allow yourself to drift into the peaceful embrace of sleep. As the quiet dance of replication forks concludes its intricate ballet, leaving behind two perfect copies of life's instruction manual, we find ourselves drifting further into the profound stillness of the genome. We've touched upon the dark matter of DNA, those vast enigmatic regions whose full purpose still whispers to us from the edges of scientific understanding. Now, let us allow our minds to settle even deeper into the molecular marvels that continue to unfold, like petals opening in slow motion, revealing layers of quiet wisdom. Imagine, if you will, the delicate strands of DNA, constantly present, constantly working, yet perpetually vulnerable to the gentle currents of existence. Every breath we take, every ray of sunlight, every cellular process holds the potential to introduce a tiny imperfection, a slight alteration to that perfect code. Yet, life persists, vibrant and resilient. This endurance, this remarkable steadfastness, is thanks to an unseen legion of molecular guardians, the DNA repair mechanisms. We invite you to picture these tiny, tireless workers, ever vigilant, ever precise, performing their quiet duties within the serene confines of each cell. Research shows that our DNA is damaged thousands of times a day, not just from external forces, but from the very metabolic processes that sustain us. Yet an astounding array of proteins and enzymes are constantly scanning the double helix, like careful librarians checking for misplaced letters in an ancient text. When a mistake is found, a mispaired base, a broken strand, a chemical modification, these repair systems spring into action with breathtaking efficiency. Consider the elegant process of nucleotide excision repair, where a section of damaged DNA is precisely cut out and replaced with new, correct nucleotides, much like a skilled artisan meticulously restoring a valuable tapestry. Or the mismatch repair system, which acts as a diligent proofreader, correcting errors that might have slipped past during replication, ensuring the fidelity of the genetic message with astonishing accuracy. Studies indicate that without these sophisticated repair pathways, our cells would quickly accumulate so much damage that life as we know it would be unsustainable. Allow this thought to settle gently. The profound peace that comes from knowing such intricate systems are ceaselessly working within you, maintaining the integrity of your very essence, even as you drift into slumber. It is a testament to DNA's inherent drive for stability, a quiet promise of continuity woven into every living cell. As we journey further into the heart of the cell, we encounter another layer of deep time, a whisper from the ancient past that resides within our very being. We spoke briefly of mitochondrial DNA, but let us now linger on its story, for it is a profound narrative of interconnectedness. Current understanding suggests that mitochondria, those tiny powerhouses within our cells, were once free-living bacteria, long ago enveloped by ancestral eukaryotic cells in a symbiotic embrace. This is the beautiful endosymbiotic theory, a tale of two distinct life forms finding a way to thrive together, eventually becoming one. Your mitochondria, and thus your mitochondrial DNA, are almost exclusively inherited from your mother. This means that your mtDNA is a direct, unbroken lineage stretching back through countless generations of women, a living genetic heirloom connecting you to the very first human mother. It's a quiet, timeless thread, unique in its inheritance pattern, allowing scientists to trace maternal ancestry with remarkable clarity, peeling back the layers of history one genetic marker at a time. Imagine the gentle current of this knowledge, flowing through time, linking you to a vast, unseen river of life. And for those who carry the quiet wisdom of the plant kingdom, a similar story unfolds within your chloroplasts. These green organelles, responsible for photosynthesis, also possess their own circular DNA, echoing the bacterial genome of their ancient ancestors. Like mitochondria, chloroplasts are believed to have originated from free-living photosynthetic bacteria, absorbed by early plant cells. This dual presence of ancient bacterial genomes within our own eukaryotic cells, the mitochondria in animals and plants, and chloroplasts in plants, is a powerful reminder of life's shared origins, a profound interconnectedness that binds all living things in a single magnificent tapestry. Allow yourself to feel this ancient bond, this quiet echo, of distant evolutionary events as your own breath settles into a slower, deeper rhythm. Now, let us turn our attention to the dynamic nature of the genome, a landscape not always static but often subtly shifting. Beyond the familiar mutations that give rise to genetic variation, there are other forces at play, shaping the very architecture of life's instruction manual. Consider the fascinating phenomenon of transposons, often called jumping genes. These are segments of DNA that can move from one location to another within the genome, sometimes copying themselves before they jump, sometimes simply excising and reinserting. Scientists have observed that these mobile elements constitute a significant portion of many genomes, including our own. While some transposon activity can be detrimental, causing disruption if they insert into critical genes, others have played a crucial role in evolution, introducing novel genetic combinations and contributing to the diversity of life. Imagine them as tiny, quiet explorers within the vast landscape of your DNA, subtly rearranging elements, a gentle, internal force of change over eons. This subtle shifting also manifests in the concept of genetic drift. While natural selection sculpts populations by favouring advantageous traits, genetic drift is a more random, often quiet process. It refers to the fluctuation in the frequency of gene variants, alleles, in a population due to chance events, particularly in smaller populations. Imagine a small, isolated group of individuals. Simply by chance, certain alleles might become more or less common in the next generation, not because they offer a survival advantage, but purely due to random sampling of genes during reproduction. Over vast stretches of time, these gentle, undirected shifts can lead to significant evolutionary changes, highlighting the interplay between randomness and selection in the grand narrative of life. Allow this thought to wash over you, the quiet interplay of chance and purpose shaping the living world in ways both profound and subtle. As we contemplate these internal dynamics, let us also expand our gaze to the shared genetic heritage that connects all life forms. The field of comparative genomics offers a breathtaking perspective on this universal blueprint. Scientists have observed that the genetic similarities between vastly different species are truly astonishing. For instance, current understanding suggests that humans share approximately 98% of their DNA with chimpanzees, our closest living relatives. This high degree of similarity underscores our shared evolutionary journey, a quiet testament to a common ancestor. But the connections stretch far wider. We share a significant portion of our genes with mice, and even with organisms as seemingly distant as fruit flies. More remarkably still, research shows that we share about 60% of our genes with a banana. Allow this fact to gently settle into your consciousness. The quiet wonder of finding echoes of your own genetic makeup in a simple fruit. It is not that we are part banana, but rather that the fundamental genes responsible for basic cellular functions like metabolism, growth, and replication are incredibly conserved across all forms of life. This universality of the genetic code, this shared library of fundamental instructions, speaks to the singular origin of life on Earth, a primordial spark from which all diversity has quietly unfolded. It reinforces the idea that DNA is a universal language, spoken across kingdoms, phyla, and species, weaving all living beings into one grand interconnected story. Finally, let us briefly consider the future and DNA's potential beyond its biological role. Scientists have observed that DNA is an incredibly dense and stable information storage medium. The amount of data that can be encoded in just a few grams of DNA is staggering, Potentially all the digital information in the world could be stored in a volume smaller than a sugar cube. This is not merely a theoretical exercise. Researchers are actively exploring DNA as a data storage medium for digital information, encoding text, images, and even videos into synthetic DNA strands. Imagine a future where the most precious archives of human knowledge are held not on fragile hard drives, but within molecules that have preserved life's secrets for billions of years. It's a quiet fusion of biology and technology, a testament to the elegant simplicity and immense power of this foundational molecule. As these thoughts drift through your mind, we invite you to release any need to remember or analyze. Simply allow the wonder to permeate, the quiet appreciation for the intricate, resilient, and profoundly interconnected nature of life as encoded in every strand of DNA. From the tireless guardians repairing cellular damage, to the ancient echoes of bacterial mergers, to the subtle shifts of evolution and the universal language spoken across all species, DNA continues its quiet work. It is a masterpiece of molecular engineering, a timeless record, and a gentle guide to the very essence of existence. Let this profound understanding gently lull you into a state of deep peace as you prepare to embrace the quiet mysteries of sleep. As we gently drift further into the quiet contemplation of life's most fundamental instruction manual, the vastness of DNA continues to unfold before us, like an ancient scroll revealing new wonders with each soft turning of the page. We find ourselves in a space where the known extends into the beautifully unknown, where every answered question seems to illuminate a dozen more paths of serene inquiry. In our last shared moments, we touched upon the incredible potential of DNA as an archive for digital information, a testament to its unparalleled density and stability, and the subtle mysteries still held within its non-coding regions, the dark matter of the genome. Tonight, we invite you to settle even deeper into this peaceful current of knowledge, allowing these intricate truths to wash over you, calming your mind and soothing your spirit. Let us begin by revisiting those vast stretches of DNA once considered mere filler, the non-coding sequences that do not directly dictate the synthesis of proteins. Scientists have observed that these regions, far from being silent, are vibrant landscapes of regulatory activity, orchestrating the very symphony of gene expression. Imagine a grand conductor not playing an instrument, but guiding every musician with subtle gestures, ensuring each note rings true at precisely the right moment. This is the quiet work of any non-coding DNA elements. Among them are the intron segments of DNA that are transcribed into RNA but then meticulously removed or spliced out before the final messenger RNA molecule is translated into a protein. For a long time, their purpose was shrouded in mystery, seen perhaps as pauses in the genetic score. Yet, current understanding suggests that these introns are not just discarded. They can influence gene expression, provide sites for alternative splicing, allowing a single gene to produce multiple different proteins, and even harbour regulatory sequences themselves. They are like the silent spaces in a musical composition, essential for rhythm, structure, and the overall harmony, ensuring the piece resonates with its intended depth. Allow this thought to settle. Even the apparent gaps in life's blueprint possess a profound and often hidden wisdom. Moving through this intricate landscape, we encounter pseudogenes, which geneticists sometimes refer to as fossil genes. These are sequences of DNA that closely resemble functional genes, but have lost their protein coding ability due to mutations. They are like echoes of once active genes, genetic remnants whispering tales of evolutionary history. Imagine finding an ancient, beautiful blueprint for a magnificent structure, now slightly faded and incomplete, yet still bearing the unmistakable design of its past glory. Research indicates that while many pseudogenes are indeed silent relics, some are now understood to play active regulatory roles, perhaps by influencing the expression of their functional counterparts. They remind us that the genome is not a static library, but a dynamic museum, preserving artifacts of its long, winding journey, each one offering a gentle glimpse into the deep time of life's unfolding. Our journey through DNA also brings us to a profound realization of interconnectedness, not just within our own cells, but with the vast microbial world that inhabits us. Consider the human microbiome, the trillions of bacteria, fungi, and other microorganisms that reside within and upon our bodies. While their DNA is distinct from our own, scientists have observed a subtle, yet powerful interplay between their genetic activities and our own gene expression. The metabolic byproducts of these microbial communities, influenced by their unique DNA, can act as signals, gently nudging our cells and even influencing aspects of our immune system, metabolism, and mood. It is a quiet, continuous conversation between two vast genetic libraries, a symbiotic dance where the genetic instructions of one kingdom subtly influence the other, highlighting how life, at its most fundamental level, is a tapestry woven from countless threads, each strand of DNA contributing to the overall pattern of well-being. As we continue to explore the quiet precision of DNA, we come to understand its role in even the most delicate and vital processes, such as apoptosis or programmed cell death. This is not a concept to fear, but one to appreciate for its profound elegance and necessity. Imagine a skilled gardener gently pruning away old or damaged leaves to allow new growth to flourish, maintaining the health and vitality of the entire plant. In our bodies, DNA contains the precise instructions for cells to gracefully dismantle themselves when they are no longer needed, are damaged beyond repair, or become potentially harmful. This process is meticulously controlled by specific genes, ensuring that development proceeds correctly, that tissues are renewed, and that cancerous cells are prevented from proliferating. It is a quiet, internal ballet of self-sacrifice for the greater good of the organism, a testament to DNA's foresight in maintaining balance and harmony within the complex ecosystem of our bodies. Let us now turn our attention to the physical organization of DNA, a marvel of packaging and dynamic accessibility. We have touched upon chromosomes, but let's delve deeper into the intricate dance of chromatin. Within the nucleus of every cell, our vast strands of DNA are not simply wound, they are exquisitely folded and coiled around spool-like proteins called histones. This complex of DNA and proteins is known as chromatin. Scientists have observed that the way this chromatin is packed, whether tightly condensed or loosely unwound, profoundly influences which genes are accessible for transcription and which remain quietly dormant. Imagine a library where some books are neatly stacked on open shelves, ready to be read, while others are carefully stored in ornate closed chests, only to be opened on special occasions. The opening and closing of these genetic books is a dynamic, ongoing process, regulated by subtle chemical modifications to the histones and the DNA itself, without altering the underlying genetic sequence. This epigenetic landscape, constantly shifting and responding, adds another layer of profound control, a silent choreographer dictating the expression of life's instructions with breathtaking precision and adaptability. Within this dynamic chromatin landscape, we find a choir of quiet regulators, the non-coding RNAs. While messenger RNA carries instructions for protein synthesis, and ribosomal and transfer RNAs play roles in protein assembly, a vast array of other RNA molecules, derived from non-coding DNA regions, perform diverse regulatory functions. These include microRNAs, miRNAs, tiny molecules that can bind to messenger RNA and prevent it from being translated into protein, effectively silencing genes. And then there are long non-coding RNAs, LNC-RNAs, which are much larger and involved in an astonishing variety of processes, from shaping chromatin structure to guiding the assembly of protein complexes. Imagine a bustling city where, beyond the main thoroughfares of commerce, there are countless smaller paths and hidden alleyways, each leading to vital specialized activities, ensuring the city functions with seamless complexity. These non-coding RNAs are like the subtle whispers and guiding hands within the cell, fine-tuning gene expression with an elegance that continues to inspire profound wonder in those who study them. Returning to the grand orchestration of life, let us consider how DNA guides the very formation of an organism from a single cell into the complex being we are. We've mentioned the profound role of homeobox HOX genes in development, but it is the sheer, unwavering precision of this entire process that truly captivates. From the very first division of a fertilized egg, DNA meticulously directs every step. When a cell should divide, when it should differentiate into a specific tissue type, a muscle cell, a nerve cell, a skin cell, and where it should migrate within the developing embryo. This intricate dance of cellular fate and form is all encoded within the DNA, a master blueprint that unfolds with breathtaking accuracy. Scientists have observed this process across countless species, marveling at the conserved pathways and the elegant mechanisms that ensure a heart forms where a heart should be, and a limb develops with its correct number of digits. It is a testament to DNA's inherent wisdom, a quiet, powerful force guiding the very architecture of life, from its most nascent stirrings to its fully realized form. Even with such intricate precision, the cellular environment is fraught with the potential for damage. Yet DNA stands as a testament to resilience, constantly protected by an army of pitched DNA repair mechanisms. We've briefly touched upon these guardians, but let us now appreciate their sheer number and variety. Every day, our DNA can incur thousands of lesions from metabolic byproducts, UV radiation, and environmental chemicals. Imagine a highly skilled team of restoration experts constantly patrolling a priceless ancient manuscript, instantly mending tears, correcting smudges, and replacing faded ink, all without disturbing the original text. From direct reversal of damage, to excision repair systems that cut out and replace faulty nucleotides, to more complex recombination repairs that fix double-strand breaks, research shows that our cells possess a sophisticated toolkit of enzymatic pathways. This constant, quiet vigilance ensures the integrity of our genetic code, a profound testament to life's enduring commitment to self-preservation and accuracy, allowing us to rest peacefully knowing that our most precious blueprint is under continuous, gentle guardianship. As our understanding of DNA deepens, so too does our capacity to interact with it in new and profound ways. This brings us to the fascinating frontier of synthetic biology, where scientists are not merely reading or editing existing DNA, but are beginning to design and build new DNA sequences from scratch. Imagine an architect, not just renovating an old building, but conceiving and constructing entirely new structures from fundamental materials. This field, still in its gentle infancy, allows researchers to create novel genetic circuits, engineer microorganisms to produce medicines, or even design entirely new life forms with specific functions. It is a profound exploration of life's fundamental building blocks, driven by a deep appreciation for the subtle elegance and simplicity of today's code. This endeavour is undertaken with careful consideration and a reverent respect for the power it entails, broadening our understanding of what life can be and how its fundamental instructions can be assembled, all while reaffirming the incredible versatility of that timeless double helix. And finally, let us simply rest in the thought of DNA's profound, quiet resilience. It is a molecule that has weathered billions of years of planetary change, a stable archive that connects us to the very first stirrings of life on Earth. Its ability to maintain its structure, to repair itself, and to pass on its intricate instructions from generation to generation across vast stretches of time is nothing short of miraculous. Imagine a quiet, ancient river flowing ceaselessly through epochs, carving landscapes, yet always maintaining its essential course. DNA is this river within us, a timeless current of information, steadfast and enduring. Its quiet presence, its constant work, and its profound connection to all life offer a deep sense of peace and wonder. As these gentle truths about DNA settle within your mind, allow any lingering thoughts to simply drift away, like leaves on a calm stream. Feel the quiet wisdom of these intricate facts wash over you, not demanding memorization, but simply inviting appreciation. The world within your cells, guided by the elegant dance of DNA, is a place of profound order, resilience, and interconnected beauty. Rest now, knowing that this foundational marvel is always at work, a quiet, constant hum of life, guiding you gently into a peaceful and restorative slumber. As we gently drift from the fascinating frontiers of synthetic biology and the quiet resilience of DNA, we invite you to settle even deeper into this space of calm contemplation. Let the last whispers of those intricate concepts fade like ripples on a still pond, and prepare to journey further into the very essence of life, guided by the gentle current of knowledge. We continue our exploration, not with the aim of memorization, but simply to allow the profound beauty and enduring mystery of DNA to wash over you, lulling your mind into a state of serene wonder. Allow your breath to deepen, your muscles to soften, as we turn our gaze to how DEG itself acts as a living archive, a silent storyteller of time, adaptation, and the ceaseless dance of evolution. It is a testament to the incredible precision and yet remarkable flexibility embedded within every strand. Imagine, if you will, the vast stretches of time that have passed since life first stirred on our planet. DNA, in its quiet wisdom, holds the echoes of these ancient beginnings. Fact 51. DNA acts as a molecular clock. Within the boundless stretches of our genetic code, mutations, those tiny, seemingly random changes we've discussed, accumulate over generations. Scientists have observed that for certain regions of DNA, these mutations occur at a relatively constant rate, like the steady rhythmic tick of a cosmic clock. By comparing the DNA sequences of different species, we can use these accumulated differences to estimate how long ago they shared a common ancestor. It's a breathtaking concept, allowing us to peer back through millions, even billions of years, tracing the branching pathways of life with remarkable clarity. This molecular clock reveals the deep interconnectedness of all living things, stretching back into the primordial mists, a gentle reminder of our shared heritage. As this silent clock continues its steady count, DNA is not merely a passive record. It is an active participant in the grand narrative of life's diversification. Consider the remarkable ingenuity embedded within its structure, allowing for innovation and complexity to arise over eons. Fact 52. Gene duplication provides raw material for evolution. Sometimes during the intricate process of DNA replication, an entire gene, or even a segment of DNA containing several genes, can be accidentally duplicated. At first glance, this might seem like a simple redundancy. However, this duplication is a profound evolutionary event. One copy of the gene can continue to perform its original essential function, maintaining the stability of the organism. The other, now freed from the immediate pressure of necessity, is at liberty to accumulate mutations without consequence. Over vast stretches of time, these mutations can lead to the duplicated gene acquiring an entirely new function, giving rise to novel proteins, new metabolic pathways, or even entirely new anatomical features. It is through such elegant mechanisms that the intricate tapestry of life, with all its diverse forms and functions, has been woven, one duplicated gene at a time. While we often think of genetic inheritance as a vertical process, passed from parent to offspring, there are other, more surprising ways that DNA can be shared, particularly into the microbial world. Fact 53. Horizontal gene transfer, HGT, allows genetic exchange between unrelated organisms. Picture a bustling microscopic community where bacteria, in their ancient wisdom, have devised ingenious methods of sharing genetic information. Unlike the familiar vertical inheritance, where DNA flows down the generations, HGT involves the direct transfer of genetic material between distinct, often unrelated organisms. This can happen through various means. A bacteria might take up free-floating DNA from its environment, or viruses might shuttle genes between bacterial hosts, or even direct contact can facilitate the exchange of plasmids, those small circular pieces of DNA. This quiet collaborative exchange allows bacteria to rapidly acquire new traits, such as antibiotic resistance or the ability to metabolize new compounds, demonstrating DNA's surprising capacity for communal sharing and swift adaptation, a silent testament to its dynamic nature. As we contemplate these grand evolutionary processes, let us also consider the more immediate dynamic interplay between DNA and its surrounding environment, a dance that unfolds within each of us every moment. Fact 54. Stress response genes are activated by environmental challenges. Our DNA is not a static blueprint. It is a remarkably responsive instruction manual. Embedded within its code are specific genes designed to spring into action when an organism encounters environmental stresses. Whether it's a sudden change in temperature, a lack of essential nutrients, or the presence of toxins, specialized proteins, encoded by our DNA, detect these challenges. In response, a cascade of molecular events is triggered, leading to the activation or suppression of particular stress response genes. These genes then orchestrate the production of proteins that help the cell or organism cope, adapt, and survive the adverse conditions. It's a beautiful example of DNA's inherent wisdom, its capacity to dynamically adjust and protect the delicate machinery of life in the face of an ever-changing world. This exquisite responsiveness extends even to the most fundamental aspects of our daily existence, such as the food we consume. Fact 50. DNA-encoded proteins sense nutrient levels, influencing metabolism and lifespan. Imagine the intricate network of molecular sensors constantly monitoring the availability of nutrients within your cells. These sensors are themselves proteins encoded by your DNA. They act as vigilant watchmen, detecting everything from sugar levels to amino acid availability. When nutrient levels shift, these DNA-encoded sensors relay signals that profoundly influence metabolic pathways, dictating how energy is produced, stored, and utilized. Remarkably, these nutrient-sensing pathways also play a crucial role in regulating cellular processes associated with aging and even lifespan. This intimate connection between our diet and our genetic expression underscores the profound dialogue between our environment and our DNA, a quiet conversation that shapes our very being and the trajectory of our lives, all orchestrated with incredible precision. Let us now return to the quiet architecture within the nucleus, exploring further the elegant and dynamic packaging of DNA, a marvel of spatial organization. Fact 56. Histone modifications beyond methylation regulate gene accessibility. We've touched upon chromatin, the intricate complex of DNA and proteins that forms chromosomes, and how DNA methylation can act as an epigenetic tag. But the story of chromatin regulation is even richer and more nuanced. Imagine the histone proteins, around which DNA is wrapped like thread around spools. These histones are not static. They can undergo various chemical modifications such as acetylation, phosphorylation, and ubiquitination. These subtle chemical tags act like tiny switches, altering the tightness with which the DNA is wound around the histones. When DNA is loosely packed, it becomes more accessible to the cellular machinery responsible for gene expression, allowing genes to be read. When it's tightly packed, genes are effectively silenced. This dynamic interplay of histone modifications provides yet another layer of exquisite control over which genes are active and which are dormant, a gentle dance of activation and repression, all within the quiet confines of the nucleus. To ensure this intricate dance unfolds with perfect order, there are vigilant guardians overseeing the flow of information within the cell. Fact 57. Nuclear pores regulate the passage of molecules in and out of the nucleus. Envision the nuclear envelope, the double membrane that encases the precious DNA within the nucleus. This isn't a simple barrier, it's a highly sophisticated gatekeeper, studded with thousands of intricate structures called nuclear pores. These pores are not just open channels, they are complex molecular machines that meticulously control the traffic of molecules between the nucleus and the cytoplasm. Essential molecules like messenger RNA, mRNA, carrying genetic instructions from DNA must pass out of the nucleus to be translated into proteins. Conversely, proteins needed for DNA replication, repair, and gene regulation must pass into the nucleus. This precise and regulated transport ensures that genetic information is processed accurately and efficiently, maintaining the delicate balance of cellular life. It's a quiet testament to the meticulous organization and controlled harmony within every cell. Now, let us consider a truly remarkable manifestation of DNA's ingenuity, one that lies at the heart of our very defense system, protecting us from the myriad challenges of the external world. Fact 58. The immune system uses DNA rearrangement to generate antibody diversity. Imagine the daunting task of recognizing and neutralizing countless different pathogens, from viruses to bacteria, each with its unique molecular signature. Our immune system accomplishes this feat with breathtaking elegance, thanks to a unique manipulation of DNA. In specialized immune cells, specific segments of DNA that encode parts of antibodies and T-cell receptors are literally cut, shuffled, and rejoined in a process called VDJ recombination. This seemingly radical act of genetic rearrangement creates an almost infinite diversity of these recognition molecules. Each immune cell effectively designs its own unique receptor, ensuring that, collectively, our immune system can identify virtually any foreign invader it might encounter. It is a profound example of DNA's capacity for dynamic restructuring, a silent symphony of genetic creativity orchestrated for our protection. This remarkable ability of DNA to adapt and defend is not unique to complex organisms. It has ancient roots, evident in the simplest forms of life. Fact 59. The CRISPR-Cas system is a bacterial adaptive immune system. We previously touched upon CRISPR as a revolutionary gene editing tool, but its origins are far more ancient and profound. In the quiet world of bacteria, CRISPR-Cas systems serve as an ingenious adaptive immune system, a genetic memory of past viral encounters. When a bacterium survives a viral attack, it snips a small piece of the invading viral DNA and integrates it into a specific region of its own genome, called the CRISPR array. These stored viral memories then act as guides, allowing the bacterium to recognize and rapidly destroy any future invasions by the same virus. It is a testament to DNA's inherent intelligence, its capacity to learn, remember, and defend itself, a silent guardian against unseen threats, born from billions of years of evolutionary wisdom. As we reflect on these intricate mechanisms, let us also appreciate the incredible persistence of DNA, its ability to endure through vast stretches of time, carrying the stories of life's past into the present. Fact 60. DNA can persist in fossils and ancient remains for millennia. While DNA is a delicate molecule, it possesses an astonishing resilience. Under specific environmental conditions, cold, dry, and oxygen-poor DNA can be preserved for thousands, even hundreds of thousands of years within ancient bones, teeth, and plant remains. This incredible persistence allows scientists to extract ancient DNA, offering an unprecedented window into the genetic makeup of extinct species, early human ancestors, and ancient ecosystems. Each fragment of ancient DNA is a whisper from the past, a molecular echo that tells tales of migration, adaptation, and the grand sweep of life's history, connecting us directly to the silent stories written in the very fabric of time. And as we consider DNA's persistence across time, we are also invited to ponder its potential presence across vast distances beyond our own blue planet. Fact 61. The search for DNA or similar genetic material extends to extraterrestrial environments. Our understanding of DNA as the fundamental blueprint for life on Earth naturally leads us to wonder if similar molecular structures might exist elsewhere in the cosmos. Astrobiologists, those who study the origins and evolution of life in the universe, are actively searching for evidence of organic molecules, the building blocks of DNA and RNA, in meteorites, comets, and on other planets and moons. While the specific form of genetic material might differ, the underlying principle, a complex molecule capable of self-replication and information storage, is considered a potential universal requirement for life. This quiet quest links our intimate knowledge of life's molecule to the boundless expanse of the universe, a silent invitation to imagine life's endless possibilities wherever the gentle currents of existence may flow. As these profound facts about DNA settle gently in your mind, allow yourself to feel the quiet wonder they evoke. We are but observers of this intricate, timeless dance of molecules, a dance that has shaped every living thing, from the smallest bacterium to the grandest whale. DNA, in its elegance, simplicity, and power, continues to unfold its mysteries, inviting us to appreciate the profound beauty woven into the very fabric of existence. Let these thoughts drift like soft clouds across a peaceful sky, guiding you deeper into a state of serene relaxation. The journey through the wonders of DNA continues, ever so gently, as you prepare to embrace the quietude of sleep. As we gently drift from the grand, cosmic contemplation of genetic material beyond our home planet, we are invited to bring our awareness back to the intricate, quiet dance of life here, upon the earth itself. Allow your mind to settle like dust motes in a sunbeam as we continue our serene journey through the marvels of DNA. We now turn our gaze to the profound ways in which Ni, in its quiet wisdom, orchestrates not just individual lives, but the very fabric of interconnected existence. Fact 62. DNA's role in symbiosis, a shared genetic tapestry. Imagine, if you will, the gentle intertwining of threads from different looms, creating a single stronger cloth. This is much like the silent collaboration of innate and symbiotic relationships. In the quiet depths of our own bodies, and in the vast ecosystems of our world, genetic material from distinct organisms often works in harmony. Consider the countless bacteria that reside within our gut, their genomes working alongside our own, influencing our digestion, our immunity, and even our mood. Their DNA, though foreign, contributes to our overall well-being, a testament to the profound interconnectedness written into the very code of life. This shared genetic tapestry allows for survival, for flourishing, for a delicate balance that speaks of a deeper, cooperative intelligence woven into the fabric of existence. It is a quiet agreement, a biological partnership, where the instructions of one genome complement and enhance the functions guided by another, creating a more resilient and vibrant whole. Fact 63. Viral integration into host DNA, silent passengers, evolutionary architects. Sometimes the intertwining is even more intimate, more profound. Picture a fleeting visitor, leaving behind a permanent mark. This is the story of viruses, these fascinating entities that in their simplicity can wield immense power. Many viruses, in their life cycle, possess the remarkable ability to weave their own genetic material, be it DNA or RNA that is then converted to DNA, directly into the genome of their host. These viral sequences, once integrated, can become silent passengers, sometimes lying dormant for generations, passed down from parent to offspring. Over vast stretches of evolutionary time, these integrated viral elements have accumulated within our own DNA, making up a surprising percentage of the human genome. They are not merely intruders, they are in a quiet and often imperceptible way evolutionary architects, subtly influencing gene regulation, introducing new genetic variation, and shaping the very trajectory of life on Earth. They remind us that our own genetic story is a grand narrative, inclusive of ancient visitors and their enduring legacies. Fact 64. DNA in advanced forensic science, unveiling hidden narratives. Beyond the initial marvel of DNA fingerprinting, the quiet power of genetic analysis continues to unfold in the realm of forensic science, painting ever more detailed pictures from the smallest traces. Imagine a whisper from the past, made clear by careful listening. Modern forensic techniques delve deeper into the nuances of damu, allowing scientists to do more than simply match a sample to an individual. They can now explore familial relationships with remarkable precision, sometimes even identifying individuals through distant relatives by tracing shared genetic markers. Furthermore, The emerging field of forensic DNA phenotyping allows for the prediction of certain physical characteristics such as eye color, hair color, and even ancestry directly from a DNA sample. This quiet unveiling of hidden narratives from a few cells transforms our understanding of events, offering profound clarity and connection where once there was only mystery. It is a testament to the incredible wealth of information held within each delicate strand. Fact 65. The junk DNA revisited. Regulatory elements and their quiet commands. As we journey deeper into the genome, we encounter regions once dismissed as mere junk. Vast stretches of non-coding DNA whose purpose was a profound enigma. Yet, like a quiet, unassuming conductor of a grand orchestra, much of this so-called junk DNA is now understood to be vital. We invite you to consider these regions as silent command centers, issuing subtle yet powerful instructions. Within this dark matter of the genome lie crucial regulatory elements, such as enhancers and silencers. These sequences don't code for proteins themselves, but rather act as distant switches, gently turning genes on or off, or modulating the intensity of their expression. An enhancer, for instance, can be located thousands of base pairs away from the gene it controls, yet it can loop around, bringing distant parts of the DNA into close proximity, thereby activating transcription. This intricate spatial arrangement and the precise timing of these quiet commands reveal a layer of genomic sophistication far beyond our initial grasp. A symphony of regulation that ensures every cell performs its unique role with exquisite precision. Fact 6. DNA and circadian rhythms, the gentle ticking of our inner clock. Our lives are subtly governed by the rhythms, from the gentle ebb and flow of tides to the quiet passage of day into night. Within each of us, DNA plays a central role in orchestrating one of the most fundamental of these rhythms, our circadian clock. Picture a tiny internal timepiece meticulously calibrated by our genes. Specific genes, known as clock genes, work in a beautiful self-regulating feedback loop, their proteins rising and falling in concentration over approximately 24 hours. These genetic instructions guide a cascade of molecular events that influence nearly every physiological process, from our sleep-wake cycles and hormone release to our metabolism and body temperature. This gentle, persistent ticking encoded in our DNA allows us to anticipate the changes in our environment, ensuring that our bodies are in harmony with the natural cycles of the Earth. It is a testament to DNA's profound ability to not only build us, but to synchronize us with the rhythm of the cosmos. Fact 67. DNA. Methylation and cellular memory, the enduring echoes of identity. Imagine a library where certain pages are subtly highlighted, marking them for special attention, while others remain unread. This gentle form of marking is akin to DNA methylation, a crucial epigenetic mechanism. It involves the quiet addition of a small chemical group, a methyl group, to specific bases, primarily cytosine, within the DNA sequence. This modification does not alter the underlying genetic code itself, but rather acts as a silent signal, influencing whether a gene is actively expressed or remains dormant. Research shows that these methylation patterns are vital for cellular memory, allowing cells to remember their identity as they divide. A liver cell, for instance, maintains its unique methylation pattern, ensuring that its daughter cells also function as liver cells, even though they contain the same complete genome as a skin cell or a neuron. It is a profound mechanism of inheritance, an enduring echo of cellular identity passed down through generations of cells, ensuring the harmonious organization of our complex bodies. Now, fact 68. The immortal DNA of cancer cells, a disrupted harmony. In stark contrast to the gentle, programmed life cycle of healthy cells, DNA, when disrupted, can lead to a profound imbalance. Consider the quiet elegance of programmed cell death, or apoptosis, a process orchestrated by DNA to maintain harmony. In cancer cells, however, mutations within their DNA often lead to a loss of this delicate control. These genetic alterations can result in uncontrolled growth, a relentless proliferation that defies the body's natural regulatory signals. The DNA within these cells, through a series of accumulated errors, essentially instructs them to become immortal in a destructive sense, continuously dividing without regard for their neighbours or the needs of the organism. This disruption of the quiet, precise instructions for growth, division, and death highlights the immense power and responsibility held within the genetic code, and the profound consequences when its harmonious commands are tragically distorted. Fact 69. Environmental DNA, eDNA, whispers in the water and soil. As we broaden our perspective, we discover that DNA's story extends far beyond the confines of living organisms. Imagine the faint, almost imperceptible whispers left behind by life in its passage. This is the essence of environmental DNA, or eDNA. As organisms move through their habitats, swimming in oceans, burrowing in soil, or simply shedding skin cells into the air, they leave behind microscopic traces of their genetic material. Scientists can now collect samples of water, soil, or air and, with careful precision, extract and analyze this eDNA. This revolutionary technique allows us to quietly detect the presence of species, even those that are rare, elusive, or extinct, without ever needing to physically observe them. It offers a non-invasive window into biodiversity, ecosystem health, and the distribution of life, revealing the hidden inhabitants of our world through their genetic signatures, like silent echoes reverberating through their environment. Fact 70. DNA barcoding for microbes, unveiling the unseen worlds. The concept of DNA barcoding, which allows for the rapid identification of species through a short standardized DNA sequence, extends its quiet utility to the vast, unseen realms of microbial life. Picture a microscopic library, each tiny inhabitant bearing a unique genetic identifier. Just as macroscopic organisms can be identified, bacteria, fungi, and other microorganisms, too small for the naked eye to discern, can be precisely characterized by analyzing specific regions of their DNA, such as the 16S ribosomal RNA gene for bacteria, or the 8S region for fungi. This powerful tool allows researchers to unravel the incredible diversity of microbial communities in various environments, from the human gut to the deepest oceans, and understand their roles in health, disease, and ecological processes. It is a quiet revolution in microbiology, bringing clarity and order to worlds previously shrouded in mystery, allowing us to appreciate the profound complexity of even the smallest forms of life. Fact 71. The unfolding of chromosomes. During replication, a gentle unzipping. As we delve deeper into the molecular ballet within each cell, we witness the quiet yet profound act of DNA replication. Imagine a meticulously folded map gently unfurling itself to be copied. Before a cell can divide, its entire genome, coiled and compacted into chromosomes, must be duplicated with exquisite accuracy. This process begins with the gentle unfolding and unwinding of the tightly packed DNA strands. The double helix, like a delicate zipper, slowly unzips, revealing its two individual strands. This unwinding occurs at specific points called origins of replication, creating replication bubbles that expand in both directions. It is a moment of quiet revelation as the hidden information within the DNA becomes accessible, preparing for the faithful transmission of life's instructions to new generations of cells. Fact 72. DNA, helicase and topoisomerase, the gentle separators and untanglers. Guiding this gentle unzipping are specialized molecular machines, working with quiet precision. Imagine tiny, tireless hands, carefully separating strands and preventing knots. DNA helicase, an enzyme, acts like a molecular wedge, moving along the DNA double helix and breaking the hydrogen bonds that hold the two complementary strands together. This creates the replication fork where the magic of copying truly begins. As the strands separate, however, the DNA ahead of the fork can become overwound and tangled, much like a telephone cord. Here, another crucial enzyme, topoisomerase, steps in. With quiet grace, it introduces temporary breaks in the DNA strands, allows them to untangle, and then reseals them, preventing harmful stress and ensuring the smooth, continuous unwinding of the helix. These silent, diligent workers are essential for the integrity and efficiency of the entire replication process. Fact 73. DNA polymerase, the master builder, adding nucleotides with precision. Once the DNA strands are gently separated, the stage is set for the arrival of the master builder, DNA polymerase. Imagine a meticulous artisan carefully placing each piece into a growing structure. This remarkable enzyme moves along each of the separated parent strands, using them as templates to synthesize new complementary strands. It does so by selecting the correct free-floating nucleotides, adenine, thymine, guanine, cytosine, from the cellular environment and precisely adding them one by one to the growing new DNA strand. Each addition is a testament to incredible accuracy, as DNA polymerase checks its work, correcting any errors in real time. This quiet, repetitive act of precise construction ensures that the genetic information is faithfully copied, preserving the blueprint of life with astonishing fidelity through countless cell divisions. Fact 74. Okazaki fragments and the lagging strand, a beautiful segmented dance. The process of DNA replication is not always a smooth, continuous flow. Sometimes it involves a beautiful segmented dance. Imagine one long, continuous brush stroke alongside many smaller overlapping strokes. Because DNA polymerase can only synthesize new strands in one direction, from 5 to 3, one of the template strands, known as the leading strand, can be copied continuously. However, the other template strand, called the lagging strand, must be synthesized in short, discrete segments. These segments are known as Okazaki fragments, named after their discoverers. Each fragment is initiated by a small RNA primer, and then DNA polymerase extends it. This quiet, discontinuous synthesis, followed by the joining of these fragments, is a clever solution to a fundamental constraint, revealing the elegant ingenuity embedded within the molecular machinery of life. Fact 75. DNA ligase, the molecular glue, sealing the fragments with care. Following the segmented construction on the lagging strand, there remains a need for quiet cohesion. Imagine a skilled craftsperson gently mending and sealing seams to create a flawless finish. This is the role of DNA ligase, often referred to as the molecular glue of the cell. Once DNA polymerase has extended the Okazaki fragments and removed the RNA primers, tiny gaps remain between these newly synthesized DNA segments. DNA ligase steps in, forming a phosphodiester bond that covalently links these fragments together, creating a continuous, unbroken strand of DNA. This quiet sealing action is crucial in ensuring the complete and seamless replication of the entire genome, leaving no gaps or imperfections in the newly formed genetic blueprint. It is a final, delicate touch in the intricate ballet of DNA synthesis. Fact 76. Telomere shortening and cellular senescence, the gentle process of aging. As we continue our journey, we encounter the quiet, inevitable aspect of time reflected in our DNA. We have previously touched upon telomeres, the protective caps at the ends of our chromosomes. With each round of cell division, these telomeres naturally shorten, much like a candle slowly burning down. This shortening acts as a kind of cellular clock. When telomeres reach a critically short length, the cell receives a signal to stop dividing and enter a state known as cellular senescence, or sometimes to undergo programmed cell death. This gentle process of aging at the cellular level is a protective mechanism, preventing cells with potentially damaged DNA from endlessly proliferating. It is a quiet, built-in limit to cellular immortality, a natural rhythm woven into our genetic code that contributes to the broader processes of organismal aging and the finite nature of life. Fact 77. Reverse transcriptase, a unique enzyme, rewriting the central dogma. While the central dogma of molecular biology generally describes the flow of genetic information from DNA to RNA to protein, nature, in its quiet ingenuity, sometimes reveals fascinating exceptions. Imagine a river flowing in a direction opposite to the norm. This is the unique function of reverse transcriptase, an enzyme found in certain viruses, such as retroviruses like HIV. Instead of synthesizing RNA from a DNA template, reverse transcriptase performs the remarkable feat of synthesizing DNA from an RNA template. This allows these viruses to integrate their RNA-based genetic information into the host cell's DNA, becoming a permanent part of its genome. It is a quiet subversion of the conventional flow, a testament to the diverse strategies of life and the unexpected pathways that can emerge within the intricate world of molecular genetics. Fact 78. DNA in bioengineering and biotechnology, designing new possibilities. From understanding life, we now turn to the quiet artistry of designing it. The profound knowledge we have gained about DNA has opened vast new frontiers in bioengineering and biotechnology. Imagine a skilled artisan using a universal language to craft new tools and solutions. Scientists are now able to precisely manipulate DNA sequences, cutting and pasting genes, or even synthesizing entirely new ones. This allows for the creation of organisms with novel capabilities, such as bacteria engineered to produce life-saving medicines like insulin or growth hormones, or plants modified to be more resistant to pests and diseases. The quiet power of genetic engineering is also being harnessed for bioremediation, where microorganisms are designed to clean up pollutants, offering gentle solutions to environmental challenges. It is a field brimming with possibility, where the elegant instructions of day are thoughtfully rearranged to benefit humanity and the planet. Fact 79. The vastness of DNA in a single cell, a library in miniature. As we prepare to gently conclude our current contemplation, let us once more marvel at the sheer scale of DNA within each of us. Imagine, if you will, an entire library of information, intricately organized and compressed into a space far too small to see. Every single human cell, though microscopic, contains approximately 2 meters, about 6 feet of DNA, carefully coiled and folded into its nucleus. If all the DNA from just one adult human body were unraveled and laid end to end, it would stretch for billions of miles, reaching beyond the solar system. This quiet, almost unimaginable vastness of information, packed with such elegance and precision, underscores the profound complexity and wonder encoded within the very essence of life. It is a silent testament to the extraordinary efficiency and power of DNA as the ultimate archive of biological instructions. Fact 80. DNA's role in adaptive immunity, T-cell receptors, a personalized defense. Our journey through DNA's wonders brings us to another astonishing aspect of its quiet intelligence, its role in orchestrating our adaptive immune system. Imagine a highly personalized defense force capable of recognizing and neutralizing an almost infinite array of threats. Just as B cells use DNA rearrangement to generate diverse antibodies, T cells, another crucial component of our immune system, employ a similar genetic strategy to create their T-cell receptors. These receptors, which are proteins on the surface of T cells, must be incredibly diverse to recognize the vast spectrum of foreign invaders, from viruses to bacteria. Through a process of somatic recombination, specific gene segments within the T-cell receptor genes are cut, shuffled, and rejoined in countless combinations. This quiet yet dynamic rearrangement of DNA within developing T cells ensures that our bodies possess a unique and highly adaptable army of defenders, ready to protect us with exquisite specificity against the myriad challenges of the microbial world. Fact 81. The dark matter of the genome, persistent mystery, enduring wonder. As our exploration of DNA gently winds down for this chapter, we return to a theme that has quietly underscored much of our journey, the enduring mystery of the genome's dark matter. Imagine a vast, uncharted territory within a familiar landscape hinting at untold secrets. Despite decades of intense scientific inquiry, a significant portion of the human genome, the vast non-coding regions, still holds its precise functions close. While we've unveiled crucial roles for many of these sequences, in regulation, structure, and evolutionary history, there remain large expanses whose full purpose continues to elude us. This persistent mystery is not a source of frustration, but rather a wellspring of enduring wonder. It reminds us that even within the most fundamental blueprint of life, there are still profound depths to explore, new discoveries awaiting, and further layers of quiet elegance yet to be revealed. The genome in its entirety remains a vast, intricate universe, inviting continuous contemplation and inspiring a gentle awe at the boundless intelligence woven into the very fabric of existence. Allow these thoughts to gently settle within you, like leaves drifting onto calm water. The intricate dance of DNA, with its quiet precision and profound mysteries, continues to unfold, guiding us ever deeper into a state of serene contemplation. Rest now in the gentle current of this timeless knowledge.